Hello everyone. So let's quickly see scientists and stains in general microbiology. First, scientists. Louis Pasteur, Pasteur Institute of Paris, pasteurization of milk, liquid media, father of microbiology, fermentation principles, autoclave theory of abiogenesis, also known as theory of spontaneous generation, which is disapproved, germ cell theory, then three vaccines, cholera, anthrax, rabies, were discovered by Louis Pasteur. Louis Pasteur is often asked as short note in universities. Another important short note is Koch postulates. So, Robert Koch, father of modern microbiology, Koch postulates Koch bacilli, cholera organisms, solid culture medium, aniline dye color, hanging drop motility. So, four postulates by Koch are constant association of causative organism with the disease, isolation in culture media is possible, growth culture inoculated in animals should produce same lesion, re-isolation from experimental animal is possible. Exceptions of Koch postulates are LPG, L for Microbacterium leprae, P for Trypanosoma pallidum, G for gonococci. Paul Ehrlich, Ehrlichia organism, father of chemotherapy, toxin antitoxin standardization, Nobel Prize, acid fast stain, which is modified Zelen stain. Then Joseph Lister, father of antiseptic surgery, which was the first person to use carbolic acid. Then Anton van Leeuwenhoek, father of light microscope, which is a kind of simple microscope. First thing visualized was animal cues. Gentian duo binocular compound microscope. Ernst Ruska, father of electron microscope. Edward Jenner, first vaccine smallpox, that is first person to use the vaccine for smallpox prepared from the cowpox. Caribou mollis, PCR. Gold standard is Sanger sequencing by Sanger. HC gram staining. Now let's see Kleinberg L forms, which were cell deficit forms. Alexander Fleming, penicillin. Barbara McClintock, transposomes, which are known as jumping genes. HCV by Michelle Huntington, Harvey J. Alter, Charles M. Rice. Then CRISPR-Cas9, which is recently discovered by chemistry scientists Emmanuel Carpentier, Jennifer A. Doudna. Then stains. So now scientists are over. Now let's see stains. Then we will do some questions. So stains fixative could either be heat fixation or it could be chemical fixation. Then stain, simple stains are methylene blue and basic fuchsin, which gives red color, and methylene blue color. Negative stain is a different kind of stain. It includes India stain and Negrosin stain. By staining the background black, organism appears white. That means we stain the background instead of staining the organism. Example is cryptococcal meningitis, which is stained in this way, capsulated and CSF fluids. Then impregnation stain deposits stain on surface of the object to make it look thicker. Silver stain is used in thinner structures like flagella and spirochetes. Levaditis stain for tissue samples, Fontana stain for fluid samples. Example of spirochetes are trypanosoma causing syphilis. Then differential stains are gram stain, acid fast stain, Albert stain. Gram stain, crystal violet, gram positive and negative, both gives purple color. Then we add iodine, which is mordant or fixative, which fixes the organism to the slide. Then decolorizer, alcohol or acetone. Gram positive gives purple color and gram negative becomes colorless on adding decolorizer. Then we add safranin red, where gram positive gives purple, gram negative gives red or pink color. Then let's do a question. Which is the most crucial step for gram staining? The step which can differentiate gram positive and negative, which is decolorizing. So, what is the concept behind gram staining? Gram positive cell wall has a more thicker layer of peptidoglycan which retains the primary stain longer, whereas gram negative cell wall has more lipopolysaccharides, making it permeable to secondary dye after decolorization, which has lipid more amount, so alcohol to acetone. Poorly gram staining organism can be remembered by mnemonic MARSIS, Mycoplasma rickettsia, Chlamydia, and Spirochetes. Zn stain, that is Ziel-Nansen stain or acid-fast stain. The procedure includes carbylfuscin stain, which gives color to the organism. Fixative is heat. Then decolorization is acid alcohol, H2SO4. Stain is methyl alcohol, Melchite green. Background appears green in color. Amount of H2SO4 varies in different organisms. For Mycobacterium TB, it's 20%. For Mycobacterium leprae, it's coccidian and coccidian parasites, it's 5%. For Nocardia and Legionella, it's 1% H2SO4. For spores and head of sperm, it's 0.25 to 0.5%. Now let us see fit for echo stain, which has 5% H2SO4, Zn stain for lepra. Then acid fast stain, which can be remembered by mnemonic my nose is cold hot. So M for Mycobacterium TB, 20% H2SO4, acid plus alcohol fast is 95%. Atypical TB acid fast. Lepra we use 5% H2SO4. Then from N, Nocardia and Legionella. From I, S, isospores, and spores are and head of sperms. We use 0.25 to 0.5% H2SO4. From C we have Cyclospora and Cryptosporidium. From H we have hooklets of Hydeae and egg of T. Saginata. Then coccidian parasite like isospore, cyclospore, and coccidosporidium. We use cold stain. Then cold Zn stain. Then we have flagella stain, which is silver stain. Lefson's and Rui stain. Dye is basic fuchsin for flagella, which gives red color. Mordant is tannic acid. Then secondary stain dye is methylene blue for cells, which gives blue color, which is remembered by mnemonic. Flagella is used for locomotion. Flagella, carbylfuchsin, fuchsin red color, L for locomotion. So name of the stain is Lefson's Rui stain. Then let's see spore stains. So spores are also stained by a stain. Schaeffer's and Fulton stain. So modified SB stain, Melchite green, gives color to the spores. Then we heat it, which is a fixative. Then decolorizer is water, and saffron in red is the secondary stain. Then Zinn-Nelson's modification, quinone stain or Gabet stain, which is cold Zn stain, where fixative is cold instead of heat. Increased phenol in carbylfuchsin, diagnosis of coccidian parasite. Albert stain. Volutin granules are present in C. diphtheria. Albert solution 1 includes toluidine blue, melachite green, and glacial acetic acid, and Albert solution 2 includes iodine. So Albert staining is done in a sequence. First we use Albert stain 1, then 2. So C. diphtheria looks like this. It has terminal granules, which are known as volutin granules, which are purple colored in this staining procedure, also known as Babes-Ernst granules, bipolar granules, and metachromatic granules. Volutin granules can be stained by Ponder stain, Loeffler's methylene blue stain. Loeffler's methylene blue is considered best stain for volutin granules. Albert stain, Neisser stain. Then volutin granules are seen along with Clostridium diphtheria, also in Yersinia yeast, Mycobacterium TB, Gardnerella, and Spirillum. So volutin granules. So these organisms can be remembered by mnemonic volutin granules. V, if we extend a line, then it becomes Y. So Yersinia and yeast. From N, if we extend one line, it becomes M. So MTB. From G, Gardnerella, and S, Spirillum. Now let's practice some questions from staining. In gram staining, the mordant is, so iodine, Lugol's iodine is the answer. We have seen that primary stain in gram staining was crystal violet, mordant or fixative was iodine, then decolorizer was alcohol, and secondary stain was safranin. Then after primary staining, the mordant has been added, but before the decolorizing agent being used, gram positive organism will appear purple, of course, but not that gram negative organism will also appear purple, because we have not added decolorizer, so both has the purple color, and after adding decolorizer, the gram negative will become colorless, and then they will gain either a red or pink color due to safranin. The next question is metachromatic granule stain are all except. So metachromatic granules or volutin granules are stained by, we have seen just now, for Loeffler's, which is best, Albert, Neisser, Ponder. So quinone is not used. Then silver impregnation method of staining is used to demonstrate thin structures like flagella and spirochetes. So spirochetes is the answer. Mycobacterium is comparative thicker. Which of the following are acid fast with 20%? For 20% we have Mycobacterium TB, which is not in the option, so M avium is correct. For M lepre we have 5% H2SO4, and for Nocardia we have 20% H2SO4, so these cannot be the answer, so most correct is M avium. For Zn staining procedure in secondary staining is, so for Zn staining we have seen that carbonyl fuchsin is primary stain, then fixative is heat, then decolorizer is acid, and secondary stain is methylene blue or Melchett green. So here the answer is Melchett green. Then the correct order for gram staining, this could be asked in Viva. So for the gram staining we have seen that crystal violet, iodine, acetone, and safranin. So that's all. Thank you.